The overall goal of this procedure is to observe and characterize pharyngeal arch movements in the developing zebrafish head. This is accomplished by first embedding a transgenic zebrafish embryo in anesthetic agarose gel. The second step is to mount the agarose-embedded embryo in a drop of methylcellulose on a microscope slide. Next, the embryo is sealed between two microscope slides with vacuum grease. Ultimately, time-lapse confocal microscopy is used to show differences between wild-type and mutant zebrafish in the morphogenetic movements of the pharyngeal arches.
The main advantage of this technique over static imaging methods is that it captures pharyngeal arch development in real time, allowing an investigator to directly observe the tissue movements that are critical to morphogenesis.
To make a bridged cover slip, superglue a 22 by 22 cover glass onto each end of a 24 by 60 cover glass, leaving the middle free. Make singles for embryos less than a day old, doubles for embryos 1 to 4 days old, or triples for embryos 5 days or older. And for each specimen, use 2 bridged cover slips. Now fill a 10 milliliter syringe with high vacuum grease. Prepare fresh anesthetic in 0.2% liquid agarose, and maintain in a 42 degrees Celsius heating block. To manually dechorionate the unhatched transgenic embryos, slowly tear the chorion open until the embryo is freed. Now transfer the dechorionated embryos into clean media containing MS-222 anesthetic.